A very good afternoon, good evening, good morning to all of you. My name is Yanling Wong, and I'm a Scientific Affairs Officer with the UNODC Laboratory and Scientific Service. As you know, I'm coordinating the UNODC International Collaborative ICE Exercises Program, or ICE program in short. So welcome to this UNODC ICE Ask the Expert sharing session. And today, sorry, please excuse me. Uh, Today, our topic is on the 2024 round of the eye structure elucidation on NPS. In this session, we will provide an overview of the laboratory performance in this eye structure elucidation round of 2024 round 3. Then we will have experts from Australia, China, and Singapore to share their experiences participating in this round. In the second session later today at 4 p.m. Vienna time, we will have experts from Spain, Canada, and Brazil to share their experience. You will find that though it is the same sample, laboratories take slightly different and innovative approaches, and we have such a great opportunity to learn from these experts. So before we start, we want to remind you that the presentations from today will be recorded and to allow colleagues who could not join us to tune in and benefit from what our speakers have prepared. By attending the meeting, you agree to the recording. The link to the private recording will be shared by email to all ICE laboratories. There will be a question and answer session after our guest presenters finish their presentations. So keep your questions till the end. And alternatively, you can also put your questions in the chat and we will read them during the Q&A. Your microphones have been disabled and will be activated in this Q&A portion. So without further ado, I would like to invite Dr. Connor Crane, Scientific Affairs Officer of the UNODC Laboratory and Scientific Services, to give his welcoming remarks. Connor, please.
Thank you, Yanling. Good morning, good afternoon, and good evening, everyone. It is my pleasure to welcome you to the fourth UNODC ICE Ask the Expert session. We are gathered here today as part of the United Nations Global Network of Forensic Laboratories. We are all committed to advancing forensic science and analytical excellence. I'd like to begin by expressing our sincere appreciation to the ICE laboratories worldwide. Your committed partnership over the past 30 years has been foundational to the success of the International Collaborative Exercises Program. Through your contributions, we have built a robust network of expertise that continues to elevate the standards of forensic drug analysis. We are also grateful to our partnership with the European Network of Forensic Science Institutes and their Drugs Working Group for supporting the implementation of a second round of this structure elucidation exercise. As many of you know, the identification of unknown NPS is one of the most pressing analytical challenges we face in forensic chemistry today. Many of these compounds often emerge with completely novel structural features, lacking reference materials or established analytical or toxicological profiles. So structural elucidation exercises provide a platform for laboratories to refine their interpretive skills, validate their analytical workflows, and contribute to a global knowledge base on emerging substances. Special thank you goes to the experts presenting today who will be sharing their insights during this session. Your expertise and methodological approaches are invaluable to the entire forensic community, and we look forward to learning from your casework, your strategy, and the innovation you used for determining the structure of these samples in this test. Let us use this session to deepen our scientific dialogue, strengthen our collaborative networks, and continually pushing the boundaries of forensic drug analysis. So once again, welcome and thank you very much for being part of this important initiative. Thank you.
Thank you, Connor. Share my slides. Before we directly go to the structure elucidation exercise, allow me to briefly give you an overview of the history of NPS structure elucidation proficiency test in NSV Drug Working Group and how we started this fruitful collaboration together. Now, seeing that there was a need to evaluate the ability of laboratories to recognize and identify even very new substances appearing in the market for which there has not been any analytical data available, the NSV Drug Working Group started to provide the European laboratories with structural elucidation proficiency testing in 2016. As you can see on this slide, the structure of the target substances NSV had designed and used in previous exercises. For the round 2022, NSV offered to partner with UNODC to extend their reach of this exercise globally. So following the success of this round, we launched another round in 2024. We would like to take this opportunity to thank NSV Drug Working Group and especially Hungarian Institute of Forensic Sciences team again, and in particular Tamas Chestregi and Tibor Vagra for the design, synthesis of the target substances and the analysis of the result. So for this kind of exercise, the target substance was custom synthesized such that it couldn't be easily purchased, and there is no available literature or database library available to compare the findings. Additionally, the substance also had to be similar to existing recent designer drugs. Laboratories were asked to identify the active substance as best they could with available techniques and provide the conclusion to each result. So the test sample of ICE round 2024-3 was provided to 34 labs from NSV and 34 labs in the ICE, total 68 labs, and this is an increase of from 51 labs in 2022. The target substance is tryptamine. Now, to disguise the identity of the target substance in the import-export permits, we use a non-trivial name, which is normally not how you would name this substance. Okay, this is the non-trivial name. So we classified the results into 4 categories. First, the correct identification of the target substance. Now, the second group is a partially correct identification, meaning that the correct substance is listed among a few possible substances. And the third category is an incorrect identification, or the laboratory is not able to identify the substance, meaning that the substance the conclusion was wrong. Yeah. And number 4 is that there was no conclusion and no substance was identified. So in general, 50 out of 68 laboratories identified the presence of a tryptamine in the sample, and 53% of the laboratories identified the target substance correctly. This includes laboratories who identified target substance directly and the laboratories who indicated the target substance among a few possibilities due to the limitation in their analytical scheme. For those who reported the correct target substance, there were 11 different names used, as you can see here on this slide. For partially correct identification, 2 labs unable to identify with confidence the actual substance, actual structure of the target substance, but they provided possible structures which included the correct structure. Now on this slide, you can see the correct structure indicated in the blue box, and then all the rest are the wrongly identified ones, and altogether there were 13 incorrect structures reported. There were also 2 laboratories who reported chemical names that could not be interpreted by OPSIN tool. Therefore, the results were evaluated as no conclusion. For future exercises, we would encourage participants to always report using the structure, this OPSIN tool. So for your convenience, we added the link on the slide. And you'll find this is a very, very useful tool. So for the next part, I would like to pass the mic to Tamas, the NSV Drug Working Group Proficiency Test Coordinator and the head of the Drug Investigation Department of the Hungarian Institute of Forensic Sciences, who will give us a deeper insight into the analysis of the results from both the NSV and the ICE labs. Tamas, over to you.
Thank you very much, Yaling. Can you show me the next slide? Oh, sorry. Oh, thank you very much. Briefly, here are the different analytical schemes or workflow used by the labs. Labs using NMR tend to be more successful in getting a correct or partially correct identification. And for successful identification, some key information was used by the laboratories. Based on GCMS and FTIR analysis, laboratories could infer that the target substance is a tryptamine. Derivatization is also a useful technique in structure elucidation. Following derivatization, GCMS analysis was able to exclude the presence of primary and secondary amine groups. Laboratories can use high-resolution MS techniques to determine molecular formula from the accurate mass. And finally, the exact position of the substituents on the indoor ring can only be determined through NMR analysis. And some laboratories face challenges in the interpretation of results and reporting. Three laboratories correctly noted their limitations in the evaluation. A few laboratories reported the structure of the target substance but did not provide sufficient analytical information to prove it. For example, a laboratory reported the methyl group on the fifth carbon atom, but this position was not confirmed by any analytical results. Few laboratories provided only mass spectral library matches for structure identification, but this approach is not reliable for structural elucidation of a new substance. A few laboratories with NMR reported incorrect structures. The reliable elucidation of substances with NMR requires suitable knowledge and experience, so laboratories should make careful interpretation of NMR spectra. And some laboratories reported the wrong isomer. We should consider all the potential isomers and confirm the results by appropriate analytical methods. In additional testing, if additional testing is not possible, it's important to indicate the possibility of other isomers in our reports. For structural elucidation of a new unknown NPS can be very challenging. There is no available information and there is no commercial available reference material on the new substance. In this exercise, the incorrect identifications of the target substance were related to several issues. Some false identifications are based solely on the use of analytical techniques not specific or sensitive for isomer discrimination. Some wrong results were based on the incorrect evaluation of differences between experimental data and the best hit found by the library search algorithm. And majority of incorrect results uh, are related to incomplete consideration of possible isomers. Therefore, laboratories that did not report the correct substance should review their analytical, SIEM, and evaluation processes. The review of identification criteria and the use of complementary technique selective for isomer discrimination is strongly recommended to reduce the risk of false identification. The cornerstone of the robust structural elucidation procedure is the consideration of all possible isomers. NMR is a useful method for structural elucidation, especially for small molecules in simple matrices. When NMR is not available on site, laboratories are encouraged to seek assistance from other institutions. Laboratories are reminded that it's crucial to cross-validate the measurement results of the different techniques. To avoid misinterpretation, it's also important to cross-validate NMR results with other techniques to detect possible discrepancies. Thank you.
Thank you, Thomas. Thank you. So we are very happy to announce that the result of this round and the related details are now available in the summary report, as you see this on the slide. The link is here, and we will also put it in the chat for you. So there is a wealth of information in this report. Not only can you learn from the exercise, but also We also have the information on the synthesis and characterization of the target substances are also included. So I strongly encourage you to have a look at this report and also to share with your colleagues. Now we come to the exciting part, the sharing of the experience from participating laboratories. So without further ado, allow me to introduce our first speaker of this session, Miss Karen Blakely. Is a senior scientist in the forensic chemistry laboratory at the Forensic Science Queensland and has over 20 years of experience in the analysis of illicit drugs. She has a particular interest in the identification of NPS and has been involved in numerous cases involving structural elucidation of novel compounds, including cathinol, synthetic cannabinoids, and steroids. So, Ms. Blakely, the floor is yours.
Thank you very much, Yanling. I will just share my presentation. Hopefully everyone can see that okay, but let me know if you can't. Hello, good morning, good evening to everyone, and thank you to the UNODC for allowing me to participate in this event. I thought I would start off by just doing a little introduction to our lab at Forensic Science Queensland. So we're a state-based forensic laboratory. Queensland has a population of about 5.6 million people across 1.7 million square kilometres. So to put that into context, we have about the population of Finland in about 5 times the area. Our forensic chemistry lab performs seized drug testing as well as clandestine laboratory examination, and we also perform a broad range of trace evidence testing. We analyze about 14,000 suspected drug exhibits per year, and most of those are street seizures and state-based operations, although we do get the occasional international border seizure. As far as equipment goes, for illicit drug analysis, GCMS is still our main tool that we use, though our reliance on LCMS-MS has grown substantially over recent years. For quantitation purposes, we're using UPLC with photodiode array, and we also have one with a single quad QDA mass detector for more complex mixtures, usually cannabinoids, FGIR, and also a GC-IRD. Some recent equipment to the laboratory that hasn't made its way into the workflow yet is a benchtop NMR and 2 Radian ASAPs. As far as our standard illicit drug identification workflow, other than our typical visual exam and documentation of the exhibit, we would usually start with reagent tests, usually Marquis and sometimes the Scott test, and then follow on to GCMS. And some exhibits will be reported on that basis. But where more testing is required, we will then use an appropriate suite of tests to meet acceptance criteria. When it comes to structural elucidation, we do take a collaborative approach. We don't have access to high-res mass spec in our laboratory at the moment, but we work very closely with our local forensic toxicology laboratory who will run some samples for us. And when it comes to NMR, as Tomas just said, we collaborate with universities. We don't perform structural elucidations routinely or regularly, but we have had a number of instances where we have performed structural elucidations, and we will always try to do it if we come across a new compound that we can't identify, because we learn so much from the process. Just a few quick examples of ones that we had done before, all the way back to 2007, before NPS was even a thing, back to 4-methyl methcathinone and N-ethylcathinone, quite a few synthetic cannabinoids over the years, including QMPSB, which is one that we did learn quite a lot from. An obscure compound, diformal mescaline, which I think has never been seen outside of Australia, to my knowledge. And more recently, we've been looking at 5-cyano-isododesnitazine, something that was identified by a CanTest drug checking service recently in Australia, and then we've also seen it in our laboratory as well. For the talk today, I wanted to just take you through our thought processes as we analyzed this sample through our standard workflow, and the changes in our belief in what the compound may be as we progressed. So we started with reagent tests. Marquis reagent and Scott reagent did not give a reaction for the compound, so we moved on to solvent extraction for GCMS. We used two solvents, an ethanol extract and also ammonia saturated hexane, which we use as a rapid base extract. Now you can see the two total ion chromatograms in the top of that slide. Both have a peak at 3.3 minutes, which is our internal standard undecane, and then a single peak at about 6.5 minutes. Looking at the mass spectrum, could see it had a base peak of 84 mass to charge, which was quite strong. It appears to have a molecular ion of 228. Unsurprisingly for this exercise, there were no good library matches, but at this point we start to think about what are the different fragments of known drug groups that might contribute a base peak of 84. So we were thinking of phenidates with a piperidine group. Amphetamines, a little less likely we thought, but still in the group, and pyrrolidino group. So for example, the nitazepines have a strong base peak of 84. Although if that molecular weight was 228, we thought that looked a little small for a nitazene. Moving on to FGIR, again, no good matches, although there are some indications of indazole type synthetic cannabinoids in that list. Again, they'd probably be larger than 228 mass. We did note a carbonyl peak present, and we're thinking potentially a cathinone may be the compound. Just to confirm whether that molecular ion in GCMS did actually reflect the molecular weight of the compound, we liaised with our colleagues in forensic toxicology, and they ran a sample for us on their QTOF to give us an accurate mass, and that reflected the closest formula was C15H20N2. So noting the absence of an oxygen there fitting with the carbonyl group that we were seeing in FTIR. We also ran the sample on our LCMSMS. Again, we try and run as many tests as we can if we have enough material, just to make sure that all of the pieces fit together, and it supported a molecular weight of 228 as well. The next test we ran was using our GC with an infrared detector. It's less sensitive than our GCMS, so we will typically run a much more concentrated extract. And as you can see from that TIC, it looks quite different to what we saw on GCMS. We still have our internal standard peak. We have our what we believe is our target analyte, alluding a little bit later, but also some broad peaks there. When we looked at the IR spectrum of our what we believe to be our target analyte, the carbonyl was no longer present, and we were getting quite a reasonable match for N-substituted tryptamines. Now, looking at where that carbonyl group, if we have a look at the broad peaks, that's where the carbonyl group is, and the best match was for pimelic acid. It's not unusual to see tryptamines as succinate salts, so we started looking at succinic acid, which wasn't in our library, but we're able to just find an IR spectrum online, I believe through SpectraBase, just to get a general indication to support we were on the right track. So at this point, we were looking at a succinate salt of a tryptamine. We went back to ATR, more for the experience just to compare, and basified a sample, extracted it into acetone, a very small volume of acetone, added that acetone to the ATR prism and let it evaporate to get a spectrum which gave the best match for N-methyl and ethyl tryptamine. So we're slowly getting closer. This led us to thinking about the compound malt, N-methyl and allyl tryptamine. So the methyl allyl amine does have a base peak of 84, and the compound has a molecular weight of 214. So 14 mass units less than the unknown compound with the same base peak. We then took a step backwards to the Ehrlich's reagent, which is used with tryptamines, and a reaction did occur. So we saw a gray-purple color form with the Ehrlich's reagent. The fact that it was reacting suggests that the indole nitrogen is not substituted because there needs to be a hydrogen there for that reaction to take place. It can sometimes hydrolyze in the reagent and you'll get a delayed reaction, but we were operating on the assumption that there was no substitution on that nitrogen. So our next step took us back to the GCMS, and we were looking at the GCMS of malt compared to our unknown. The malt GCMS has a 130 mass to charge fragment associated with the tryptamine. We have a 144 mass to charge fragment, which indicates the addition of CH2 to the indole or the replacement of hydrogen with a methyl group. For those very observant people seeing the original TIC who notice a very slight blip on the baseline, going back to have a look, we also saw the presence of butane dioic acid, also known as succinic acid. So again, that's further supporting a tryptamine with the succinate salt. Based on that data, we came to a point of looking at tentative structures, and without NMR, that probably would have been as far as we went. And we would have said that we believe there was an addition of CH2 or replacement of a hydrogen with the CH3 on the indole, either on the ring or next to the nitrogen, and that we believe there was a C4H8 substitution on the nitrogen with some possible structures there. But we were able to collaborate with Griffith University, and we were getting very close to when the submission was due at that point. So we didn't get to do a full review of the report, but had enough data to put forward the structure that we believed it was. And we noted in our report that if it was for evidential purposes for court, that we would have still required further in-depth review of that NMR report. I think the talks today were generally about advice to be given to labs. I would say that every time we've undertaken a structural elucidation, the laboratory as a whole has learned a lot from it. So it's a very valuable process to undertake, which can sometimes be difficult when you are a high throughput forensic laboratory to take the time. Our strategy is a little bit of a shotgun approach of trying to generate as much analytical data as we can to reveal pieces of that puzzle, and generate tentative structures, which we can then search in literature and try to either confirm or exclude. So thank you very much.
Thank you. Thank you very much, Ms. Bakely, for the very, very interesting presentation. For most of us, we do not have a wide range of equipment like you, so it's good to see your collaboration with academia and other laboratories when there are instruments that you don't have in the laboratory. So this is something that we can learn from. So thank you very much. So moving on, we'd like to introduce then our next two speakers, Ms. Chui Mei Liu and Mr. Bo Yang Xu, from the National Narcotics Laboratory of the Drug Intelligence and Forensic Center of the Ministry of Public Security in China. So firstly, Ms. Liu is a senior scientist and has over 16 years of experience in the area of forensic drug analysis, and her major research areas are in drug and NPS analysis, drug profiling, quantitative NMR, and spectral techniques. She will be presenting on the overview of the NPS landscape and identification protocols in mainland China. So Mr. Xu is a senior scientist with over 9 years of experience specializing in fields of forensic science and NPS analysis. He has participated in several scientific research projects aimed at developing analytical techniques and methods for the detection and characterization of new and emerging substances. He will be sharing their experience in this round of structural elucidation of NPS. So I'd like to pass the time to Ms. Liu. Over to you, please. Ms. Liu, you're muted. Yes, we can hear you. And we
Can you
can
hear
see your
me?
presentation. Yes, very good. Yeah,
Okay,
please
thank
go
you,
ahead.
Lily. Good morning, good afternoon, and good evening, distinguished colleagues from all over the world. I'm Tumi Liu from National Narcotics Laboratory, Drug Intelligence and Forensic Center of Ministry of Public Security, China. Today, I'm very glad to have this opportunity to give you a brief introduction of the overview of the NPS landscape and identification protocols in mainland China. For my introduction, first, NPS trend in 2024-2025. Next, identification protocols of NPS. From July 2024 to July 2025, nearly 3,000 suspected samples have been analyzed by our lab, National Narcotics Laboratory, and 5 regional subcenters. This table shows the top 12 NPS detected. Etomidate is the most frequently detected substances. In addition, isopropoxate, etomidate, 3-fluoroetomidate, isobutomidate, 4-fluoroetomidate, 2,6-dichlorine 3-fluoroetomidate all belong to etomidate analogs. This indicates that etomidate analogs are now the most frequently abused NPS category in China. Tylotamine, 2-oxo-PCPR, 2-oxo-PCE, and 2-FXE all belong to 5-sectin-type substances, indicating that 5-sectin-type substances are now the second most frequently abused NPS category in China. In July 2025, lysine analogs, as well as 12 other substances, were placed under control in China, covering the recently detected NPS after July 2024. For the control list, it includes 7 etomidate analogs, 2 5-sectin-type substances, 1 synthetic casinols, 1 semi-synthetic cannabinoids, and 1 other substances. Lysine analogs was put under the generic control for the according to the definition, lysine analogs refer to derivatives of etonisine with structure modification on the benzomethyl group, the methylene group, the phenyl group, and the amino group. This slide shows the chemical structures of newly detected lysine analogs. For example, the nitrogen-nitrogen dimethylaminoetonisine, methylene dioxylnisine, nitrogen diethyl methylene dioxynisine, ethylene etonisine, 5-cyanoisodesnisine, and proton desnisine. And these nisine analogs are most frequently abused in the form of herbal blends and with concentration of 0.1% to 7%. This slide shows the chemical structures of the regulated etomidate analogs, including the 3-fluorine etomidate and 3-butomidate analogs, 4-fluorine etomidate, APB7, and 2,6-dichlorine 3-fluorine etomidate. And these etomidate analogs are most frequently abused in the form of e-cigarette oil samples with concentration of 0.69% to 7.7%. This slide shows the chemical structures of 5 other regulated NPS. For example, including the 3-methoxy-PCPY, 2-oxo-PCPR, nitrogen isopropyl butylone, thiophantamine, HHC. And these substances are mainly abused in the form of powder, liquid, or capsule. For the second part, our lab has established an analytical procedure for the analysis of suspected NPS samples. First, appropriate solvent was used to extract the sample, and then the prepared solution is analyzed by GCMS and LCMS. If there's a peak detected, then the spectrum of the peak is searched within the library, and after the library searching, special attention should be paid to the presence of potential positional isomers. If the answer is yes, then we will use the NMR or FTIR or superfluorescent chromatography to analyze the sample to identify the isomer. If the library search fails to match any of known substances, it indicates that there may be an unknown sample. Then we will perform the NMR analysis to elucidate its structure. Sometimes there may be no peak detected by the GCMS and LCMS. And in that situation, we will use the FTIR to analyze the sample. For some compounds, for example, like the inorganic compounds or carbohydrate compounds, they cannot be detected by the GCMS and LCMS, but can be detected by the FTIR. And following the above identification protocols, we can finally get the result. This slide shows the GCMS method we used for our routine NPS analysis. DB5 mass column is used. For the column oven program, 100 degree was held for 3 minutes and then increased to 320 degree at 20 degree per minute and held at 320 degree for 15 minutes. The total runtime is 25 minutes. The earliest elution compound is 2FA with retention time of 2.51 minutes, and the latest elution compound is EG2201 with retention time of 23.41 minutes. And for the GCMS, we use different libraries. We use both the internal library and also the external library. For the internal library, we have already established a library that contains more than 500 drugs, NPS precursors, and other substances. And for the external libraries, we use the NIST library, SW drug mass library, the Kaiman spectrum library. And you can download these libraries from the website. And this the SW drug and Kaiman library are updated regularly. And this slide shows the UPLC Q12MS method that we used for the analysis of NPS. A bifinal column or is used, and the total runtime is 15 minutes. For the 446 target compounds, the earliest elution compound is BZP with retention time of 2.12 minutes, and the latest elution compound is EG18 with retention time of 11.23 minutes. For the unknown compounds, we will perform the NMR analysis to acquire the protein NMR, carbon NMR, fluor NMR, carbon depth, as well as COSY, HSQC, HMNBC, and noisy data. By combining the LCMS, GCMS, and also the NMR data, we can finally elucidate the structure of the unknown substances. Sometimes we need to quantify the compounds that we don't have the reference material. Then we will use the QMR method. For more information about the structure elucidation or the QMR and analysis method, you can reference to our published materials. That's all for my introduction, and thanks for your attention. Next, my colleague Boyang will give you more detailed information about the analysis of the ICE test samples.
Okay. Hello, everyone. I'm Boyang Xu from the Ministry of Public Security, Drug Intelligence and Forensic Center, National Narcotic Laboratory Zhejiang Regional Center. I'm delighted to be here today to share our experiences regarding the ICE structure elucidation of NPS round 2024 third. Now I will dive into the work of analyzing NPS by drawing on our practical experiences. Regarding the analytical scheme, we adopt a three-step method to elucidate the structure of this sample. First, we utilize the FTIR and the HRMS to rapidly determine the relevant structures, which have the chemical formula C15H20N2 and contains functional groups such as methylene group, methyl group, and phenyl group, and amino group. Next, we relied on NMR to clarify the atomic connectivity and combine the GC mass to verify fragment information. Finally, we cross-checked all data sets and confirmed this structure. First, we analyze the unknown samples by FTIR, the instrument setting ATR mode, scanning time 16, resolution 4 wave numbers, scan range 4,000 to 400 scan wave numbers. As a result, no match was found in the database. So we analyzed the spectrum. Each peak in the table corresponds to a specific moiety, indicating the presence of amino, methylene, and also substitute aromatic groups. Next, let's examine the UHPLC HRMS data. Instrument parameters was set as follows. Observe this target peak at 229.16971. We can by using this tool, we obtain a molecular formula fit of C15H21N2 for the compound. Then we use the GC mass to analyze this sample. Instrument parameters was set as follows. Also, no best match found in the database. Based on this mass spectrum, we have deduced the possible fragmentation pathways of this substance. Finally, we characterize the unknown by NMR. Standard instrument parameters were used. We talk about 10 milligram dissolved in DMSO, hydrogen NMR, carbon NMR, depth 135. COSY, HS, QC, and BC spectra was acquired for the sample. In order to assign chemical shift to the proposed molecular structure, the NMR spectrum shows impurity peaks consistent with organic acid. Based on experiences, we propose that impurity is likely succinic acid. As a result, structure elucidation was achieved by comprehensive analysis, allow for complete assignment of all signals. Okay, the ICE sample differs from routine analysis. In our daily work, the samples we encounter are typically mixture with target analysis, usually present at low concentration, which requires further enrichment and purification. However, in this case, the NPS has a high purity and thus do not need enrichment and purification. In regular casework, we usually need to elucidate the structure of unknown substance while dealing with cases involving NPS or unknown chemical substance, but these situations are not common. However, with the continuous emergence of NPS, the demand is on an increasing trend. For these cases, we usually need to conduct a detailed chemical analysis of the unknown substance to determine their composition and structure, thereby providing key evidence for solving the cases. The main challenge we faced was that the targeted substance wasn't in our existing database, which meant we had to use a comprehensive multi-technique analytical workflows. This process took a lot of time and careful work to make sure the structure was accurate. Also, when interpreting the NMR data, the substance was in a short form with organic acid impurities, leading to a clear interference peaks in both carbon NMR and hydrogen NMR spectra. The issues made the initial spectral assignment tricky, but we figured them out by examining the spectra and cross-checking the data, which let us successfully determine the structure. During our lab's participation in this test round, I think several key facts contribute to our success. First and foremost, we have a team of experienced professionals with solid theoretical knowledge and extensive practical experiences in analyzing and elucidating the structure of NPS. Our expertise allows us to quickly and accurately develop an analytical protocols for complex samples. Secondly, our lab is equipped with state-of-the-art instrumentation that provide precise and reliable data, enabling high precision structure elucidation. Our HRMS delivers high-quality data for accurate molecular formula determination, while our NMR provides detailed structure insights, significantly enhance our analytical accuracy and efficiency. Moreover, we have established a comprehensive quality management system. Throughout the test process, we strictly follow this system from sample receipt to result reporting. However, despite our satisfactory performance in this test round, we recognize that the speed of data analysis should be improved. Moreover, we lack the ability to access existing knowledge and information database. To enhance the speed and accuracy of data analysis, we need to further strengthen our team members' training in data analysis method and technique, improve our capability in big data and artificial intelligence predictive analysis so as to improve work efficiency. Okay, finally, here are some advice. Firstly, focus on team building. Structural elucidation of NPS is high technique and requires a professional with backgrounds in chemistry and pharmacy. Labs should cultivate relevant professionals, provide them with good learning environment, and encourage continuous learning. Secondly, invest in necessary instruments. The instruments are essential for structure elucidation. Labs should configure instruments like HIMS, NMR, FTIR, and the GCMS based on their needs. Furthermore, establish a quality management system. Quality is vital for a laboratory. Detailed operating procedure and quality control standards should be developed to ensure high quality results. Lastly, enhance internal training and knowledge updating. The fields of NPS structural elucidation is constantly evolving. Labs should organize regular technical training and academic lecture. Encourage personnel to follow academic journals to stay updated with the latest research and methods. In summary, with adequate preparation in personnel, equipment, quality management, and training levels can achieve outstanding results and contribute to combating NPS abuse and criminal activities. Okay, that's all for my sharing today. Thank you for your time and attention.
Thank you. Thank you to you and Ms. Liu for the very, very informative presentation. So thank you, Mr. Xu, for your advice to the labs, what to do when considering integrating NPS structuralization into your normal work. And also to Ms. Liu, we appreciate the sharing of the new trends of NPS in China and also your analytical methods and all the publications for us to learn from. Thank you so much. So moving on, last but not least, let us introduce our final speaker. Ms. Larissa C. is a scientist at the Illicit Drugs Laboratory of the Health Sciences Authority, Singapore since 2019, where her primary role is in the forensic examination and analysis of seized drug exhibits. She is a core member of the specialized team for investigation of NPS and contributes to the development of new methodologies for the elucidation and identification of NPS. She has experience in mass spectrometric techniques such as GCMS and LCMS, where she was involved in method development and validation. Ms. C, the floor is yours.
Thank you, Yanling. I'll just share my slides. Okay. Hi, everyone. Thank you for today. I will be sharing my experience on the eye structure elucidation PT that was conducted last year, as well as to share some insight into our laboratory's approach to NPS analysis. To give some context, in recent years, our laboratory typically detects about 5 to 10 new NPS a year, of which 1 to 2 may require full structural elucidation. Hence, it helps us to have an established workflow for clarity on which main techniques we will use initially, as well as what options are available for further investigation. Here I share the techniques that we use in our analysis. Routine analysis usually involves Raman for screening of certain sample types, for example, eyes, ketamine, powders, or ecstasy, followed by other techniques such as GCMS, LC, and TLC if needed. Our laboratory benefits from having an expanded range of techniques available to aid us in structural elucidation of unknowns. These include NMR, LCMS, LCHRMS, and GCIRD. The reason why we employ this range of techniques is because different techniques have different strengths. So, for example, GCMS, NMR, and crystallography provide us with detailed structural information, while LCDAD, LCMS, and LCHRMS are valuable non-thermal techniques. We also utilize GCIRD and FTIR to differentiate between isomers and PrEP-LC and PrEP-TLC for the purification of unknowns. I will now introduce our laboratory's workflow for the analysis of NPS. So suspected NPS are first detected by GCMS analysis. If reference material is available, the reference will be analyzed with the sample for retention time comparison and mass spectral matching. Analysis using a non-thermal technique is also performed in order to confirm the molecular mass of the analyte and ensure that the analyte detected in the GCMS was not a product of thermal degradation. When there is no reference material available and the identity of the analyte is not confirmed, the analyte detected will be referred to the NPS team for follow-up action. The NPS team is a group of about 8 officers who specialize in the analysis of NPS. They have an expertise in the analysis and interpretation of a range of analytical data, which allows them to work effectively to elucidate structures and identify novel NPS. Additionally, in the first detection of any new NPS in our laboratory, regardless of whether reference material is available, the NPS team will conduct a comprehensive investigation and review to confirm the identity of the drug detected. While investigating an unknown substances, if purification is required, the NPS team member will purify the sample using these few techniques. And upon isolating the compound, further analysis will be performed using a range of techniques in order to confirm the identity of the NPS. If purification is not feasible due to insufficient sample or complex matrix, and the identity of the analyte cannot be confirmed, intelligence will be provided to the law enforcement agency. So in this PT, the sample was first analyzed using GCMS analysis. Purification was not required, and LC-HRMS and NMR were the techniques that we used to confirm the identity of the unknown substance. I will now bring you through our analysis and the key findings that led us to conclude the structure. So the small peak matched to succinic acid, suggesting that the substance was a succinate salt. And the main peak matched to these two structures indicated on the left. These were entries that were available in the commercial library. And the fragmentation along the bonds indicated certain fragments that overlap with the mass spectrum that we obtained for this sample and gave us clues to what fragments might be part of the structure of the unknown. Specifically, the fragments with the mass to charge ratio 84 suggested a pyrrolidino ethyl group, and the mass fragment with mass to charge ratio 144 indeed suggested an indole group with a methyl attached to it, and the mass fragment 115 suggested there was an indole fragment. Through LC-ORBI trap analysis, we also confirmed the molecular mass of the structure as well as the molecular formula. However, we required other techniques to confirm the position of the methyl group. In order to obtain specific information about the structural connectivity, 1D and 2D NMR experiments were performed using deuterated DMSO as a solvent. We performed COSY, HSQC, and HMBC in particular, as they provide valuable information of through-bond correlations. So NMR enabled us to confirm a few things. Firstly, that there was a pyrrolidine structure in the compound, that there was no substitution on the benzene ring of the indole, and also that the methyl substitution was on the C2 of the indole structure as indicated here. However, an unexpected observation was made. We expected to observe coupling between the two H10 protons and the two H11 protons. However, we observed in the NMR spectrum a singlet that corresponded to an integral of 4. This suggested that there was no coupling between the protons, and the 2D HSQC data indicated that the 4 protons were bound to 2 different carbon atoms with 2 different chemical shifts. So this led us to question whether the proposed structure was incorrect or whether there might be something going on with the NMR data obtained. The sample was hence re-prepared in deuterated methanol, and the same set of NMR experiments were performed. This time, the H10 and H11 protons were observed as 2 sets of multiplet peaks, and there were correlations observed in the COSY experiment. So we suspect it is possible that there was some interaction of the analyte with the solvent that caused the appearance as a singlet in DMSO. So these NMR experiments, we confirmed that this was the structure of the unknown compound. On reflection of our experience in this round of PT and the NPS workflow of our laboratory, I'll be sharing some of our laboratory's considerations in the approach for NPS analysis and structure elucidation of unknowns. Firstly, when there may not be a reference available in literature or commercial mass spectral libraries, we find that close matches to the entries in the libraries can provide some clues to similar structures or mass fragments of the unknown. Hence, it is beneficial to have a range of commercial libraries available in our laboratory. Some of the free libraries that are regularly updated and which our laboratory uses are the SWIGDRUG and SWI and Cayman Spectral Library. Secondly, different techniques provide different information, so understanding the limitations and advantages of the various techniques allows us to obtain clues which enable us to piece the puzzle and deduce the structure of an unknown substance. Another consideration in the analysis of unknown NPS is that the stability of the substance is unknown. This includes the pH stability considerations, for example, when doing basic extraction, or also thermal stability when GCMS is used. Hence, appropriate analytical instruments and sample preparation is key, and using non-thermal techniques to complement the analysis workflow is beneficial. The final consideration that I would like to highlight is the complexity of NMR analysis. For instance, if the sample is impure or a salt, there's a need to determine which signals are from the compound of interest. The choice of solvent can also have an impact, for example, for differences in solubility, for better resolution for identifying structural or positional isomers, or as observed in example here, when different splitting pattern might be observed in different solvents. Therefore, analyzing the sample using 2D NMR experiments or with an alternative solvent can provide additional information and aid structure elucidation. Finally, thinking a bit bigger picture, I would like to share our organization's approach to NPS response and strategies. The first would be to enhance staff competency for officers to be familiar with NPS analysis and the use of different analytical techniques, including non-routine ones. To enhance technical capability, this could include exploring new techniques for the laboratory or using orthogonal techniques. Next, something to consider would be to enhance the workflow. For example, having dedicated teams that are familiar with NPS analysis and structure elucidation, or also establishing SOPs for new drug identification, investigation, and reporting. To enhance knowledge, to stay updated with information that is shared by other labs or through early warning systems, and by attending conferences. And finally, to engage stakeholders or counterparts through partnership to share information on new NPS detections and drug trends. For us, this includes labs within HSA, such as the Analytical Toxicology Lab, or labs overseas as well. We also work quite closely with the law enforcement agency for appropriate response and legislation. So I've come to the end of my sharing. Thank you for your kind attention.
Thank you, Miss C, for the very, very insightful presentation. I appreciate your last slide quite a lot, highlighting that this really not just about the equipment that you have in your lab, and also the collaboration with academia for NMR, etc. But it's also equally important to invest and build up and enhance staff competency, knowledge, and technical capabilities to be able to do structure elucidation. So thank you so much once again to all our speakers. Now, without further ado, allow me to introduce our first speaker of this session, Mr. Jesus Mahugo Castuera is the head of the drug department of the Customs and Excise Central Laboratory in Spain. Sorry. Yeah, 1 minute. Yeah, please. While I introduce you, sorry. And he has over 20 years of experience in analysis of illicit drugs, NPS, and their precursors. He is the Spanish customs delegate and representative in numerous technical committees, networks, and and international organizations in the field of controlled substances, chemicals, biological agents and elements, as well as equipment for the manufacture of dual-use chemical and biological substance. So, Mr. Castuera, the floor is yours. Thank you.
Thank you very much for allowing me to be here and share with you our experience and a reflection on our participation in the IC structural elucidation of NPS test. My presentation will briefly discuss the significance of customs and Spanish customs laboratories and our interest in participating in inter-cooperation trials dedicated to the structural elucidation of organic compounds such as the new psychoactive substances, the NPS. International trade is the exchange of goods and services across national borders, allowing countries to access products, resources, and technologies that may not be able domestically. These interconnected systems contributed to economic growth, cultural exchange, and the development of global markets. However, the movement of goods between countries requires oversight to ensure legality, fairness, and safety. For this reason, control borders exist, managed by customs authorities. Customs controls all over the world involves the inspection and verification of goods entering and exiting a country. This process includes checking documents and performing physical inspection. To support these functions, customs laboratories play an essential role. These laboratories perform scientific analysis on goods to verify product authenticity, classification, composition, and compliance with health, safety, and environmental standards. For this reason, we analyze any product that you can imagine. In Spain, we have 4 customs laboratories: Sevilla, Valencia, Barcelona, and the Central Laboratory in Madrid, where the main staff and most of the important instruments are located. We belong to the National Tax Agency. With regard to drug trafficking, customs is the first point of control for drugs entering here in the European Union. However, Spanish customs are not competent judicial authorities. They are the Ministry of Health and the Ministry of Justice. Large consignments are usually sent directly to these agencies, and we only analyze them if there is suspicion that they may contain drugs, usually cocaine. It is usually postal parcels or small shipments that we receive out of laboratories in Madrid and Barcelona to determine what they are. Quantification of these items is rarely carried out. As you can imagine, we receive a wide unknown powder that we need to identify. It could be anything, from a simple harmless product such as flour, plastic, starch, etc., to a pharmaceutical product, a drug, a precursor, or a precursor like aging agent, or even the latest NPS. Anything. Therefore, the structural elucidation of organic compounds is a task that we need to develop and practice because there have been many occasions when we have had to use it to guess the true nature of an unknown product. Here are some examples of compounds that we have had to roll up our sleeves to figure out the chemical structure. The same applies to the challenging world of NPS. Here are the 6 new structures that no one had found before and which were therefore reported to the European Early Warning System for the first time. Fortunately, Spanish customs have excellent material resources at its disposal, and sufficient resources have been allocated to the drugs department. However, the same cannot be said for staffing, as we only have 2 people to analyze a significant number of samples each year. When chemists attempt to determine the structure of a compound, they rely on analytical techniques such as nuclear magnetic resonance and infrared spectroscopy, mass spectrometry, or ultraviolet visible spectroscopy. Each of these methods provides unique pieces of information like fragments of a puzzle learning how to interpret these data and combine them into a coherent structural model requires practice, reasoning, and patience. It's not enough to simply know the theory. A chemist must be able to apply the knowledge to real experiment results, which are often imperfect or ambiguous. For all these reasons mentioned above, it's obvious that we are very interested in participating in any trial that tests the ability to determine unknown structure, such as that proposed by the United Nations IC program, who has fully understood these needs. And we began with our sample problem. The first thing we usually do is take an infrared spectrum of the sample. At first glance, we can infer that we have a band at 3277 could correspond to the mean. The one at 2400 could belong to a salt, and the one at 1704 to an ester or an acid. Okay. The NMR proton in the deuterated dimethyl sulfoxide indicates 10.8 ppm, one proton belonging to an acid or a secondary amine. The aromatic part of the spectrum shows an orthosubstitute benzene ring structure. Also, at 3 and 1.9 ppm, there are 3 multiplets of 4 proton each. Additionally, in the 2.3 ppm region, we found 2 singlets with 4 and 3 proton each. It is certain that the singlet with 3 protons is a methyl group. We now have carbon NMR in the form of an APT experiment, that is, odd carbons, odd carbons, methane and methyl carbons are found at the top, and even carbons, quaternary and methylene groups at the bottom. The signal at 175 ppm indicates probably an acid or ester. The aromatic part, highlighted in red, together with the information provided by the nitrogen-15 HMBC experiment and the band in the infrared at 3277 centimeters minus 1 of the infrared spectrum defines a disubstituted indole structure in the nitrogen ring. The methyl groups appears in the carbon-13 experiment at 12 ppm. By analyzing the aliphatic part, we discovered several methylene groups, several of which are even equivalent. This leads us to think of a structure of the pyrrolidine or piperidine type. The COSI experiment confirms the disubstituted indole structure in the nitrogen ring. Similarly, the correlation marked in red, together with information obtained for the nitrogen-15 HMBC experiment, confirms the pyrrolidine moiety. In the HSQC experiment, one of the multiplets of 4 hydrogens in the 3.0 ppm region is due to the 2 different carbons. Therefore, we have an ethylene moiety in the target molecule. Let us take a moment to recap all the information we have now. What pieces of the puzzle we have figured out so far? Here they are. Now, let us try to combine them and see if they are reflected in the experimental data. The methyl could be one of the substitutions of the indole part, and indeed it is. In the HMBC, the 3 hydrogens of the methyl singlet interact with 2 of the aromatic carbons of the indole. The correct position of the methyl is confirmed by the interaction by the hydrogen at 11 ppm, the NaH of the indole, and the methyl at 3 bonds. In other words, the methyl is close to the nitrogen of the indole. This arrangement is also confirmed by the interaction observed in the 15 nitrogen HMBC. The 2 found marked by 2 blue arrows can only be interpreted if the methyl is in the position closest to the indole nitrogen. On the other hand, what if we connect the ethylene part to the position led by the indole once we have connected the methyl? Well, the multiplet of 4 hydrogens at 2.95 ppm belonged to the 2 carbon SO3 interactions in the HNBC experiment with the aromatic carbon. Therefore, it is connected that way. Similarly, the same multiplet shows interactions with another fragment that was not yet connected, the pyrrolidine residue. Right, now we have a possible extractor. Let's check it with another technique. How does it behave with the result of gas chromatography? We have in the chromatogram only one peak that shows 3 main fragments. 228.3, that is the molecular weight of the proposed structure, 1441 and 842.2, which are the masses of the most stable structure resulting from the breakdown of our molecule. Coming back to the NMR experiment, we detected a significant correlation in the HNBC between the 2 remaining CH2 and the carbon of the 175 ppm, the acid of the ester. Our experience and a little inspiration led us to believe that this could be due to a structure such as that of the succinate acid. We corroborated this hypothesis by performing an injection in a HPLC device with a refractive index detector prepared for the analysis of organic acids in wines and fermented beverages that we have in our laboratory. We obtained only 1 peak in the retention time of the succinate acid. We had it. Now we had to see whether the succinate acid was in the form of 1 or 2 salts. Taking advantage of the fact that the protons in resonance have a signal proportional to the numbers, we could see that for every 2 methylenes groups in the succinate acid, there was only 1 methyl group in the molecule found. We concluded that only monosuccinated of the indole compound was present. However, the question now was whether the carboxylic acid remaining in the succinate was in acid form or in sodium form. With a simple sodium 23 NMR experiment, we saw that there was no sodium signal, so we concluded that the succinate acid was in the monoacid form. Therefore, to summarize, the structure hidden behind the intercomparative test was the 2-methyl, 3,2-pyrrolidine, 1-eel, ethyl, 1H, indole monosuccinate. In summary, we can conclude that this structural elucidation is a complex process that requires a great deal of practice and therefore personnel dedicated specifically to it. It also involves having many instrumental techniques available at the same time in order to have most, if not all, of the pieces of the puzzle that will enable his final resolution. Similarly, structural elucidation is a necessary procedure in many laboratories, such as customs laboratories, and must be taken into account in the planning of intercomparison tests by entities engaged in this activity to meet the needs of checking the technical competence of laboratories. Thank you very much for the opportunity given to me in the UNDCIC Laboratory Network to share our experience. Thank you.
Thank you. Thank you so much, Mr. Castuera. It's for the very, very important, informative presentation. For me, it's very interesting to see you start off with IR and NMR, and then you use then the GCMS and HPLC refractive index, to confirm. Yeah. So I thought that's very interesting, and thank you for sharing that with us.
Thank you.
So, yeah, thank you. Next, I would like to introduce our next speaker. Ms. Lily Liu. She has been a member of the Health Canada's Drug Analysis Services since 2018, and she holds a master's degree in organic chemistry and is actively involved in innovative projects that leverage cross-cutting edge NMR instrumentation, and her work focuses on multi-drug quantitative measurements and the structural elucidation of new and emerging substances found in drug samples. Additionally, she is also active in sharing and developing innovative approaches with partners across Canada and globally. I'd like to pass the time to Ms. Liu. Over to you, please.
Thank you. Hi, everyone. Thank you so much for the opportunity to share our experience in structure elucidation. My name is Lily Liu, as mentioned, and I'm a chemistry specialist at the Strategic Research and Scientific Development Group at Health Canada. So since many of you may not be too familiar with us, I will briefly introduce our organization structure and the service channels that our group provides within Health Canada. From there, I'll go into the analytical process and workflow for samples that come through our lab and share some of the NPSs that we've elucidated using our analytical workflow over the past fiscal year. And I'll specifically discuss how we've approached the last round of the ICE elucidation. And then finally, I'll wrap it up by sharing some of the factors that I think contributed to our success during this round. So controlled substances are analyzed within Health Canada in the regulatory branch, specifically within the Drug Analysis Services, or DAS. DAS identify controlled substances listed in the Controlled Drugs and Substances Act, and they report their results in accordance to this act on what's called a certificate of analysis. They also share statistics and trends based on the submitted samples. There has been many changes in the way Health Canada has approached the opioid crisis, and one of those changes is reflected in the Canadian Drugs and Substance Strategy, which led to the incorporation of substance use services and supports with compassion, equity, comprehensiveness, and collaboration as the core values. So with that in mind, our group, Strategic Research and Scientific Development, was born under DAS, as they have been building experience and expertise in drug analysis. So our group not only support the R&D in DAS, but we're also building collaborative relationships with drug checking and harm reduction groups across Canada. And we do this through a few different channels, our biggest one being confirmatory testing, which is where we provide drug checking and harm reduction groups with high quality laboratory testing to identify and quantify as many as the components, as many components in the sample as we can. This is different from what DAS does, where the certificate of analysis identifies and sometimes quantitate compounds that are listed in the CDSA. So the difference between the two, the difference between the two would be for confirmatory testing, we will try to tell you exactly what's in there, including the diluent and how much. We also differentiate between isomers. So for example, ortho, meta, or para-fluorophenol, for DAS, they would report it under the same item, but for us, we specifically identify the isomer. This is because the purpose is to collaborate with harm reduction and drug checking groups to help increase their analytical capability. And in order for them to understand the limitations and benefits of their technology, they must also understand exactly what they're looking at. So confirmatory testing gives a much more comprehensive and in-depth picture of the drug analysis. And we are a small but mighty group across Canada, and we service over 15 different groups. Some also manage a network of groups within their own network. And beside that, using our experience and knowledge, we're also sharing our experience in drug analysis and general analysis with our clients on an ad hoc basis. We are also launching the training sample program. It's a little bit like the ICE program, but within Canada only and for our harm reduction partners. So similarly, we will make homogeneous samples of unknown composition for our partners, and then who will then do the test on their own, and through this have another data point to evaluate their program. So through the confirmatory analysis over the years, which we started in 2018, this chart goes all the way to the end of 2024. And the light green line represents the complexity of the samples over time. So overall, the trend has been going up. And even during around COVID times, while you can see that the number of samples analyzed has slowed down due to various reasons, the complexity still remained quite high. And after a small dip in 2022, it's been constantly on the rise ever since. This is in part because of NPSs or new psychoactive substances, but also from the fact that there are a lot of mixing of drugs now. Fentanyl aren't often seen by themselves, but are often seen in combinations with benzos, nitazines, and so on. And so with this complexity, we had to develop an analytical process that can capture different drug classes at different concentration, and of course, remain accurate. So one of our main rules is that we always try to use 2 analytical methods to issue a result of a drug. We don't need 2 methods to identify lactose, however. So the workflow goes something like what you see on the screen. We begin with a screening method on the GCMS and perform a limit of reporting check using GCFID. We try to be as accurate as possible, and we can report down to approximately 0.1% weight by weight by fentanyl and 0.2% weight by weight for benzos and nitazine. So depending on the concentration and the sample composition, we try to run most things on the NMR, where we are able to identify and quantitate at the same time. However, if the nature of the sample does not allow for adequate NMR analysis, then we will use the QTOF or IR or both at the same time. The QTOF is great for very low concentration samples that are also very small in quantity. In drug checking, it is sometimes quite difficult for the service users to give a lot of their samples. So we may get anywhere between 2 to 5 milligrams, so quite small. And so through all of these instruments that we use the DASIS library, and we also use download libraries from Caymans, Swift Drugs, CFSRE, and so on. And for the most part, most of the compounds we encounter are caught through this analytical workflow. If not, sometimes we will have to go digging to look at scientific literature, such as patents, journal articles, and available monographs from well-known organizations. And finally, if it's a truly unknown that we haven't encountered before, then that's when we excitedly start the structural elucidation process. This requires the acquisition of the analysis that were already in the workflow, so the GCMS, high resolution mass spec, which is the QTOF, infrared spectroscopy through either GCIR or ATR, and the additional step, which is the 1D NMR analysis of unknowns. So in the last fiscal year, our group have elucidated a total of 23 different structures. 16 were from our clients, otherwise known as the non-traditional client, NTC. That's the drug checking and harm reduction groups, and 7 were requests from DAS. So on this slide here, you'll see that there is an LSD analog, a synthetic cannabinoid, some stimulants, and 5 different tryptamines. We also see a lot of new nitazines, benzos, and some precursors. And it's also very interesting to me how regional NPS and drug analysis is. I imagine not all of these are new to you and maybe even be common in your region. So all that is to say, we use the same analytical technique or scheme on our ICE structural elucidation. And for the GCMS, we were able to get a base peak of 84 and M plus at 228. There are no isotopic pattern that we can see associated with bromine or chlorine. It is possible that the compound was degraded during this process and we're not seeing the M plus, but for now, we don't have a reason to doubt that. So we're assuming it is quite a small molecule. Unfortunately, the compound was not suitable to run on our LCMS method. So at the time, we decided to move toward NMR to see if we can crack this case without high resolution mass spec. So this is probably something that I haven't really heard people discuss too much about when it comes to NMR analysis of unknown, which is that picking the right solvent can really make your life a lot easier or a lot harder. So in my first attempt, I ran the sample in chloroform, and when you zoom in here, you can see that the aliphatic region contains 4 distinct peaks, 3 of which shows peak broadening, elements of second order coupling, especially the one around the 3.5 ppm area. Since the protons are broadened, it is likely that the carbons that are attached to these protons are also broadened, and therefore the 2D correlation might be difficult to observe. Plus, if you look at the integration, I know it's a little bit hard to see, we're seeing 9 different protons in only 2 different chemical shifts. So this will make differentiating between them and figuring out the structure of the compound quite difficult. So instead of continuing on doing analysis on this solvent, I tried a different one. The second attempt was with DMSO, and this is the opposite of improvement, and it actually got quite worse. Peak broadening is still observed, and now the 2 singlets that were well separated are not anymore. And if you look at the other 2 peaks in green, there are now 12 protons that are under the 2 chemical shifts. So the peaks are even less resolved. Luckily, my third attempt was a charm, and we ended up with quite a decent resolution. While there are peak broadening still observed, there's still 6 different peaks that are more defined. And this was the solvent that I used to run my carbon and 2D analysis, such as HSQC, HMBC, and H2BC. Sorry, this is a little bit brighter here. So the HSQC gives you 1 bond correlation between the carbon and protons that are attached to it. And then from there, using HMBC, which gives a 3 or 4 bond correlation, this gives you an idea of how these protons and carbons are connected to each other. And by just working through them slowly with some trial and error, the structure and the connections start to reveal itself. It is also through HMBC that confirms that there might be 2 components in the sample. So this brings me to the factors of success during this elucidation. One is through pattern recognition. So the indole ring core, which is diagnostic of tryptamine, you may recall from the previous slide, we elucidated quite a bit of those last year. The proton coupling pattern is quite recognizable. There's also recognition for the behavior of the aliphatic protons as well. Methylene and heterocyclic protons can undergo conformational changes in solution, causing broadening of peaks. And so while problem solving, I'm also thinking that this compound will probably have a methylene and a cyclic amine. This behavior is most often observed in fentanylogs and precursors as well in some solvents. And the second thing is to pick the right solvents. So to ensure that the aliphatic protons are easily observable and aren't in conditions where it's moving faster than the NMR timescale, resulting in peak broadening, which then causes the per carbon peak to also be hard to observe, and then the correlation may not be appearing. I also want to point out the usage of H2BC experiments. This was new to me a few years ago, and now is one of my favorite experiments to use during elucidation. So HMBC gives you 3 or 4 bond correlations, which is quite comprehensive, but H2BC filters those out and share 2 bond correlations. That's illustrated here on the far right. So this can be very helpful when you have a complex compound. It can be hard to figure out which HMBC correlation is 3 or 4 bond away. And so this helps narrow down the possibilities, making your elucidation a lot quicker and easier. Finally, I want to also point out that the lack of evidence is also still a piece of the puzzle. So as I previously mentioned in GCMS, the lack of isotopic peaks indicated a lack of chlorine or bromine, and that's important to keep in mind as well. And finally, the peer review process is one of the reasons I love working with my team. We're naturally quite curious people, and we're very rigorous with each other. We're not afraid to challenge each other. And so part of the peer review process is that we ensure that we come to the same conclusions separately and that we agree with each other. And that concludes my presentation on this year's UNODC ice samples. Thank you so much.
Thank you. Thank you very much, Ms. Liu, for the very, very insightful presentation. I can see how important it is to have experience using NMR and interpreting it. And also thank you for showing us your persistence in your endeavor to find the right solvent. I think now so far we have not heard, how difficult it is to find the right solvent. Thank you for showing that to us. So last but not least, let me introduce our final speaker of today, Mr. Adriano Otavio Maldena is a forensic chemist at the National Institute of Criminalistics of the Brazilian Federal Police. With over 25 years of experience and a strong interest in drug analysis, chemical profiling, structure elucidation, spectroscopic characterization, and industrial applications, Dr. Adriano plays a key role in the analysis and characterization of illicit drugs seized by the federal police throughout Brazil, providing critical insights and expertise to support criminal investigation. Mr. Baldeda, the floor is yours.
Thank you very much, Yanling. It's really a pleasure to see you again. And I'd like to begin. Thank you for everything in UNODC and Dr. Tamash for your tricky molecules that you always send to us. It's a really pleasure to try to solve that puzzle. But it's our job, It's so we are always really happy to talk with the UNODC audience because we understand we are in a developing country and we know that sometimes we don't have all the equipments or all the facilities to try to solve an interesting exercise like this. Well, let's begin. Just a little bit about our institution. Also, this is the National Institute of Criminalistics here in Brasilia, Brazil. I live here since 2003. I came from the South Brazil, but now I live in Brasilia. It's in the center of Brazil, is the Brazilian capital. This is our institute. It's not only forensic chemistry institute, but all the disciplines in criminalistics, like financial, ballistics, DNA. It's a Disney World of the forensic analysis in Brazil. It's our federal police National Institute of Criminalistics. So we have several areas, and we try to work together. We have in the chemistry lab, I'm going to show a little bit about this, but we also have equipments that we can use, like the X-ray diffraction or electronic microscope. As I saw in the Spain customs, we have access also, but now in our laboratory, but in our friend's laboratory nearby. Fortunately, we didn't use those for this exercise. Well, we received the sample. The sample has a code. We insert it in our quality assurance system. It's very important to us to participate. It's a way to show the capacity of the laboratory throughout our quality management system. So we start all our analysis starts dissolving the sample. Usually it's a white powder, so we use a lot of methanol. This has advantages and disadvantages, but it's how we try to see to start, and it was soluble in methanol. The solid material we run in the FTIR also. We saw the same bands that Ms. Liu pointed, some NH, probably a salt here, and carbonyl, but no hits in the libraries. And that's always a problem for those kind of analysis. We run also in our portable Raman, and again, no hits. Of course, there were some not so good hits there that lead us some clues. And I always try to understand this kind of exercise as a puzzle, as Ms. Lu already pointed that. And we have to join the pieces. And the interesting part is we don't have to start from the same point, from the same starting point. You can solve the final puzzle from different approaches. That's the beauty of this kind of exercise in my point of view. So we usually to start our approach injecting the sample in the GCMS. So I did it. So it was a single pick, a really nice pick in almost 6 minutes in our method. And there's a good match actually, more than 90, almost 90% of similarity with an indol here. And okay, so it's a good result. You can see here good correlation between peaks. You know, almost everything is here. And okay, a designer drug library, and the methyl group is here, is in the aromatic position, and not here in the nearby the nitrogen, as we already know is the right place. And that's the lesson that we have in this kind of situation. The GCMS is it's the most used, in my point of view, in forensic chemistry analysis. And of course, it can provide some very misunderstanding results, and we have to be aware. That's why this kind of discussion is important. Of course, so we have this, a nice peak, and a nice GCMS, a nice mass spectra with a nice hit. Everything is done. But as we know, NPS can be really tricky. We can have thermal instability, we can have isomers, we can have mixtures. So when we are dealing with something that didn't give us a good match in the IR or the Raman, and it's a very easygoing thing in the GCMS, we have to open our eyes to another techniques. That's something is wrong here. Of course, we also have some sample preparations. We use a lot of derivatization for GCMS analysis. And this was a nice way to find the salt form, because we check that the same peak, the same major peak that we noticed at first was not derivatized, so it was already there. So no derivatization, but we derivatizate the acid, succinic acid. So that's our way to find which was the salt form. So the succinic salt possible. And of course, we have a history already. We participate in 2002 exercise, and there was a fumarate salt. So we are starting to know Tamasa enough to understand that he likes those organic salts. So, okay, could be a succinic salt. We have, fortunately, since 2023, so 2 years ago, we installed a new NMR equipment in our lab, a 600 megahertz Bruker equipment. And with that, we of course, we start before using the university for this kind of problems or this kind of analysis. But now we have our own equipment in Brasilia. And this is the only NMR equipment in Brazil in a forensic chemistry laboratory. So we try to integrate this equipment with another forensic service in our country. Well, I chose methanol because I already know that methanol was okay for for the crude sample. So we choose methanol, deuterated methanol, as the solvent, and we had a quite clean and straightforward NMR proton analysis here. So when we look at this, we can check here the aromatic we check all the succinic salt signals out here, like here, the CH2. We check also the proportion as Mr. Mahugo did. We check that was one succinic for one molecule of the base, and this help us a lot to try to mount that puzzle that we have here. And if we check here, is almost the same molecule, but the attribution that we are going to propose has the methyl group not here in the aromatic position, has the GCMS proposed, but in that position nearby the nitrogen. Okay, so that could be the difference. The carbon, it's always very important. Here we check also the carbonyls from the succinate, and for this kind of molecule, we check that we have the same the right number of carbons. And of course, there's a lot of ways to interpret this, but I'm going to a little bit different approach because I'd like to show you. So with the hydrogen, carbon, and 2D analysis, it's not so difficult to understand this part of the molecule. The question that is tricky, in my opinion, is exactly the position of this methyl group. So when we perform, for example, a COSY analysis in the NMR analysis, we can check that the aromatic protons only can be a system that has this position with no substitution here. Because if the substitution was here, the pattern would be different. If the methyl group is not here, this hydrogen would be a singlet or a very narrow peak. So this didn't happen here. So as Ms. Liu said, no result or a result that you don't see, it's also a result. So this kind of information that can provide us like this is a zoom of the COSY. We perform also the HMBC for connection between the aromatics, and we can see that all aromatics are nearby each other here. So there's no methyl group in any position in this moiety. Okay, so with this we finish, and in the first saw the attribution. So the exercise was done, but we tried a little bit more. We convert the sample to a base form. So we dissolve the sample in water, add some drops of base, extract with ether, ethyl ether, combine the extracts, dry it with sulfate, manganese sulfate, and filtrate, evaporate, and dilute and dissolve, in deuterated chloroform. And we conduct almost the same experiments. And the results was the same for the same structure. But one experiment was really suitable in my point of view for this NMR. It's the noisy, 1D noisy. The noisy is that NOE is the nuclear overhouse effect, where the NMR signal of one nucleus changed the intensity of a neighbor. So if we irradiate one signal, we, by this NOA effect, we can check who is the hydrogen that is close to that moiety that we irradiate. So for example, we irradiate here the methyl group, and we check that if we irradiate the methyl group, there are some increases here that are not connected with the aromatic group. So the methyl group has to be far away from that part of the molecule. So it's another way to show that this methyl group can't be here. It has to be in the other side, in the other side of the molecule. If we irradiate, for example, these methylene groups here in the pyrrolidine, we only see the increase of these neighbor hydrogens. So it's coherent with this kind of attribution. And when we irradiate this aromatic, we only see increases in these two aromatics and also DCH2s here in the middle of the molecule. So it's all connected. So you can go step by step checking if your attribution makes sense or not with these NOE experiments. I really enjoy this kind of approach. So, of course, at the end, we perform a QTOF, LC QTOF, because once an expert from NMR told me, what's the first NMR experiment? Is to check the molecular formula, because sometimes it's tricky to find nitrogens, for example. So is the number of nitrogens correct here? Because sometimes we have nitrogens in the NMR has difficulties to identify those nitrogens. Not difficulties, but not the ordinary experiments will show them. So we have this, we perform the HPLC MS QTOF, and we have a very straightforward result showing the molecular formula for a protonated analysis. Okay, so going to the end, for me, the strategical analysis of NPS is like, like the astronomers does. They can choose different wavelengths to observe a galaxy like this. The radio is like this. You can use infrared, visible, ultraviolet, X-ray, many ways to see the same galaxy, but the results has to converge to a final result that makes sense to every tool that every light that you use to analyze that galaxy. We have the same issue with the NPS. We have to perform a lot of different analysis, and all of them has to converge to a final result. So that's my point of view. I hope you enjoy. I'd like to thank you to Gilberto Aquino, is my colleague in the NMR analysis and the NPS identification here in Brazil. Of course, our staff in UNODC, special hello to Connor, Yanling, and Romana that are always very kind to help us to present these results for you, and of course for my institution, the Brazilian Federal Police. Thank you a lot. Muito obrigado em português.
Thank you very much, Mr. Maldena, for the very, very interesting presentation. Thank you for showing us how you use the derivatization step, to find more clues. And also, it's also interesting that you convert the sample back to base and to just test again and see if you missed out anything. Thank you for showing us that. And I especially like your last slide. It summarized the main purpose of our structural elucidation exercise. We sincerely hope, that all participants enjoyed putting together all these pieces of the puzzles using whatever different equipment you have in the laboratory, or even collaboration with academia, coming, to a conclusion. So we hope this exercise was a good learning experience to you all, as much as it is a good experience for us organizing it.